Welcome to the Deep Dive. Today we're taking a close look at lymphangioleiomyomatosis, or LAM for short.
Right. LAM. It's a rare lung condition, affects mostly women.
And what strikes me is how long it often takes to actually get a diagnosis. We're talking years sometimes.
Yeah, the average is, well, it can be upwards of 4 years. And that delay, it really tells you we desperately need better ways, better tools to spot it earlier.
Exactly.
Mhm.
And for you listening, maybe you want to get a good grasp of medical topics, get the info quickly, but, thoroughly.
Mm-hmm.
Well, this deep dive should help. We're going to explore some recent, pretty interesting research into what could be new clues for LAM.
And we'll do it without drowning you in all the super technical jargon. The idea is to understand the research, not get bogged down.
Right. So our mission today, break down a study that looked at alternate proteins in the blood serum of women with LAM.
They use a technique, quantitative proteomics, basically a way to precisely measure lots of different proteins.
But we're going to focus on what they found, the key findings, and really what it could mean for understanding LAM.
Exactly. So if proteomics sounds a bit intimidating, honestly, don't worry about it.
Huh.
We'll walk you through the important bits, why these discoveries might be, well, potential game changers.
Okay, great. Let's dive in then. This study, it was published in PLOS One. What was the main objective?
So the researchers, their goal was pretty clear. Find proteins in the serum, that's the liquid bit of your blood, right?
Right.
Find proteins there that could act as biomarkers for LAM.
Biomarkers, like early warning signs in the body.
Precisely.
Yeah.
Finding the right ones could mean, faster diagnosis, more accuracy, and maybe even a better handle on how LAM actually develops.
So how did they go about finding these potential biomarkers?
Well, what they did, it's quite interesting. They took blood serum samples from women who have LAM and they pooled them together.
Pooled them.
Yeah, combined them. And then compared that pool to a similar pool of samples from healthy women matched for age.
Okay. And how did they compare the proteins?
They use this technology called ITRAQ. It lets them quantify, basically measure the amounts of many different proteins really precisely, see what's up, what's down. And then, importantly, they didn't just stop there. To double-check, to validate, they used another method called ELISA for some of the key proteins they identified. They tested those in a larger set of individual patient samples.
Right, so validation adds confidence. So after all that detailed work, what was the headline finding? What did they uncover?
They pinpointed 14 proteins. 14 that showed significantly different levels in the LAM patient serum compared to the healthy controls.
14. And when you say significant, what does that mean practically?
It's a statistical thing. They use a measure called a p-value. If it's below 0.05 like it was here, it basically means there's a low probability that these differences are just due to random chance. So we're pretty confident these differences are real.
Okay, 14 proteins. Now, here's where I think it gets really interesting. You mentioned some of these relate to the extracellular matrix, the ECM.
That's right. Several of them are involved in ECM remodeling. The ECM, you can think of it like the scaffolding, the structure that holds our tissues together.
Okay, the scaffolding.
And these proteins are like the construction crew. Some build it up, some break it down. The study found some really notable changes in a few key crew members.
Let's talk about those examples. Fibronectin was one, right?
Yes, fibronectin. So in the blood serum of the LAM patients, they found about a 30% decrease, less fibronectin floating around. And that was statistically significant.
Less in the blood. Okay.
But, and this is the fascinating part, when they looked directly at lung tissue from LAM patients, they actually found increased amounts of fibronectin deposited there compared to healthy lung tissue.
Whoa, hang on. So less in the blood, but more stuck in the lung tissue.
Exactly. They confirmed that buildup in the lung tissue using a technique called immunohistochemistry.
So the scaffolding material isn't circulating properly, it's getting trapped in the lungs. That seems like a key piece of the puzzle. What could that imbalance mean?
It's a critical question, absolutely. And it wasn't the only protein showing this kind of pattern. They also looked closely at von Willebrand factor, or VWF.
Okay, Vera, what do they find there?
Similar story, actually. A significant reduction, around 40% in the blood serum of LAM patients.
40% lower in the blood.
Right. And again, when they used that immunohistochemistry technique on the lung tissue, they found more VWF staining, especially in those typical cystic areas you see in LAM lungs.
So VWF might also be, aggregating or getting stuck in those damaged lung regions?
That's what the findings suggest, yeah. Like it's being pulled out of circulation and accumulating where the cysts are forming.
So that's two key proteins, lower in the blood, higher in the lungs. Did any go the other way, higher in the blood?
Yes, one of the validated ones did, kallikrein III. This one showed about a 25% increase in the serum of LAM patients.
An increase. And what about in the lung tissue itself?
Also increased levels in the LAM lung tissue, confirmed by immunohistochemistry. And interestingly, they also found more signs of reactive oxygen species, ROS, or oxidative stress in those same areas.
ROS. That sounds like it could contribute to damage.
It could, yes. Kallikrein III is known to be involved in breaking down that ECM scaffolding, and it might also promote the production of ROS, so it could be part of the disease process itself.
You know, I read somewhere kallikrein III is also used as a biomarker for prostate cancer.
That's right, it is. PSA, prostate-specific antigen, is actually kallikrein III.
That's unexpected. The same molecule playing roles in such different conditions. Does that hint at anything, do you think? Any shared pathways?
It really just underscores how complex biology is, doesn't it? These proteins can wear multiple hats. The last of the 4 main validated proteins was plasminogen.
Plasminogen, and that was up or down in the blood?
That one was decreased in LAM serum, which actually fits with some earlier research that had shown more plasminogen staining specifically within the LAM lesions in the lung tissue.
Okay, let me try and summarize this for myself and for everyone listening. For the 4 main proteins they double-checked, fibronectin, VEGF, plasminogen, all down in the blood, but seem to be accumulating in the lungs.
Correct.
But kallikrein III is up in the blood and also seems to be up in the lung tissue itself, potentially linked to ECM breakdown and stress.
You've got it. That's the pattern for those 4 key ones.
A really interesting kind of complex pattern. Did they identify others beyond those 4?
They did. Remember, there were 14 total that showed significant differences in the initial ITRAQ screen. The study listed the others too, even though they weren't validated with ELISA in this particular paper.
Okay, what were some of those?
Well, there were several that were decreased in LAM serum. Heparin Cofactor II was one, sex hormone-binding globulin, SHBG was another. Also, LPS-binding protein and fetuin B were lower.
So several more showing decreased levels. Any that were increased besides kallikrein III?
Yes. Apolipoprotein A was increased, and other apolipoproteins as a group showed higher levels. Phospholipid transfer protein, PLTP, that was up. And talon 1 was also increased in the LAM serum.
Wow. Okay. That's quite a list of proteins showing altered levels. When you step back and look at all 14, what's the bigger picture? What does this collection of changes tell us about what might be going wrong in LAM?
That's where the pathway analysis comes in. The researchers looked at how these proteins normally function and interact, and a really important theme emerged.
Which was?
12 out of the 14 proteins they identified are known to be involved in 2 key processes. Cell trafficking, basically, how cells move around the body, and again, that ECM remodeling, the building and breakdown of tissue structure.
Cell trafficking and ECM remodeling. Why are those particularly significant for LAM?
Well, think about LAM. You have these abnormal, smooth, muscle-like cells that seem to migrate to the lungs and then disrupt the normal lung structure, creating cysts. That involves both cell movement, trafficking, and extensive changes to the tissue scaffolding, the ECM.
Ah, I see.
And it might also help explain something quite challenging about LAM, why the disease can sometimes recur even after a lung transplant.
Right, because if these processes involving circulating proteins are happening systemically throughout the body, just replacing the lungs might not fix the underlying issue completely.
Exactly. It suggests these are deep-seated, body-wide processes, not just confined to the lungs.
That's a really crucial insight. Now, here's where we connect this study to some other information, maybe a bit unexpected. You mentioned looking into other connections for these biomarkers.
Yes. This wasn't part of the original PLOS One study's goal, but when you look at the broader medical literature, something quite fascinating pops up.
There you go.
Many of these same proteins, the ones altered in LAM, also show altered levels during various parasitic infections.
Parasitic infections, that seems completely unrelated.
On the surface, yes,
Huh.
but the overlap is quite striking when you start digging into it. It raises this really interesting possibility.
Yeah.
Maybe understanding how parasites affect these proteins could give us new angles on LAM itself.
Okay, that's definitely surprising. Let's explore that link. You went through the proteins one by one, starting with fibronectin decreased in LAM serum. How does that connect to parasites?
Well, it turns out several different types of parasites, things like Leishmania, Trypanosoma cruzi, Trichomonas vaginalis, even Entomoeba histolytica, have been shown to either reduce fibronectin levels or mess with how it functions.
Yeah. Like, do the parasites break it down?
Some seem to, yeah, or they interfere with how our own cells bind to it. It seems to be a strategy some parasites use.
Interesting. What about VWF, lower in the LAM serum, higher in the lungs? Any parasite link there?
Malaria is the main connection that comes up. Plasmodium parasites, the ones that cause malaria, can really disrupt the whole blood clotting system, and that includes affecting VEGF levels, sometimes lowering them in circulation.
Okay, malaria for VWF and kallikrein III, the one that was elevated in lamb serum.
So, while the LAM study looked at kallikrein III, there's research showing increased levels of a closely related protein, kallikrein IV, in schistosomiasis.
The schistosomiasis. That's caused by parasitic worms, right? Blood flukes.
Exactly. And these parasites are known to mess with the body's kinin system, which could potentially lead to changes in kallikrein levels.
Right. And the last of the validated ones, plasminogen, which was down in lamb serum.
Again, yes. Parasites like Plasmodium falciparum, the main malaria culprit, and Leishmania mexicana, they can both lead to lower plasminogen levels.
Why would they do that?
It seems they actually hijack the system. Plasminogen gets converted to plasmin, an enzyme that breaks down proteins. Parasites can use this plasmin to help them invade tissues or even hide from our immune system.
Wow. Okay, so that's overlap for all four of the validated proteins. What about the others identified in the LAM study? Does the connections keep going?
They really do. It's quite remarkable. For instance, think about the overall balance of protein-digesting enzymes. We saw kallikrein III was up in lamb. Well, broadly speaking, many parasitic infections trigger an increase in serine protease inhibitors molecules that block those enzymes. So there's a general theme of protease imbalance.
Okay. Any others?
What about VEGF-D? That's a pretty well-known marker, often elevated in lamb, right?
Yeah, used in diagnosis sometimes.
Well, guess what? Increased VEGF-D levels have also been linked to parasitic infections like malaria and filariasis. So even that established lamb marker has this potential parasitic connection.
That is striking. What about SHBG, sex hormone-binding globulin, decreased in lamb?
Yeah, there's a known link between parasitic infections generally and hormonal imbalances. The immune response itself, the inflammation, the signaling molecules released during an infection, they can all impact the endocrine system and potentially affect SHBG levels.
Okay. And heparin cofactor II, also lower in lamb.
Yep, decreased heparin cofactor II has also been seen in malaria, again pointing to disruptions in the blood clotting and anti-clotting balance during infection.
Which is pretty consistent. What about the apolipoproteins? They were increased in lamb.
Right, and parasites like Trypanosoma brucei, Plasmodium, Leishmania, Entamoeba histolytica, they've all been associated with changes in apolipoprotein levels. Some parasites seem to scavenge cholesterol and lipoproteins from the host, maybe even use them to help evade the immune system.
Immune evasion. Interesting. And Fechuin B, which was decreased in lamb.
Reduced Fechuin B is sometimes seen in parasitic infections that cause a lot of inflammation and tissue damage, like schistosomiasis and fascioliasis, which is liver fluke disease.
Makes sense. Phospholipid transfer protein, PLTP, increased in lamb.
Also linked to malaria, Plasmodium falciparum specifically, the parasite messes with lipid transfer mechanisms.
And LPS binding protein, LBP, down in lamb.
A reduction in LBP has been seen with parasitic worms like Fasciola hepatica. They seem to release stuff that interferes with LBP's job in the immune response.
Okay, almost there. Lastly, Talon 1, which was increased in lamb.
Talon 1 is involved in inflammation, getting cells to stick together, and cell movement, all processes that are heavily involved in the body's response to various parasitic infections, malaria, trypanosomiasis, leishmaniasis, toxoplasmosis, the list goes on.
It's actually quite incredible. For basically all 14 proteins identified as potential LAM biomarkers, there's a documented link, in some context, to altered levels during parasitic infections.
It is a very striking co-occurrence, isn't it? Now, it's crucial to stress again, the original LAM study wasn't looking for this connection at all.
Right, it was focused purely on identifying LAM biomarkers.
Exactly, but the fact that these changes overlap with what's seen in these other conditions, especially parasitic diseases, well, it certainly raises some fascinating questions for future research, doesn't it?
It really does. It makes you wonder, could there be some underlying, maybe environmental factor or even a subtle infectious trigger involved in LAM that we just haven't considered?
It opens up avenues of thought, for sure.
Okay, let's pull back slightly to the direct implications for LAM patients right now. Remind us, what are the current standard ways LAM is diagnosed and treated?
Diagnosis usually relies heavily on imaging, specifically a high-resolution CT scan HRCT of the chest looking for those characteristic lung cysts.
But that's not always definitive.
Not always, no. Sometimes other conditions can look similar on a scan, so a lung biopsy might be needed to be certain. We mentioned VEGF-D testing in the blood earlier. That's becoming more common as a less invasive biomarker, which is great.
But you said it's not available everywhere.
Right. For example, it's not readily available as a commercial test in Australia currently, so there's still a real need for more accessible, reliable biomarkers.
In treatment, what's the main approach?
The primary drug therapy currently is sirolimus, or sometimes everolimus. These drugs target a pathway called mTOR, which is known to be overactive in LAM cells.
And does that cure it?
It doesn't cure it, but it can help stabilize lung function and slow down the progression of the disease for many patients. But, like many medications, it can have side effects.
Which reinforces the need for maybe different approaches.
Absolutely. We need a better, deeper understanding of the fundamental mechanisms driving the disease, which could lead to more targeted, potentially less toxic therapies. And that's where understanding these protein changes could be really valuable.
So wrapping up the findings from this specific proteomic study then, the big takeaway for you listening is that it's given us this novel set of potential biomarkers for LAM.
Exactly. These 14 proteins, particularly the four they validated, fibronectin, VWF, kallikrein III, and plasminogen, they offer promise. Promise for potentially earlier, maybe less invasive diagnosis down the line.
And beyond diagnosis.
Beyond diagnosis, understanding why these specific proteins involved in tissue remodeling and cell movement are altered, that could give us really crucial insights into how LAM actually develops and progresses. And that hopefully could open doors to new treatment strategies.
Well, this has certainly been a fascinating deep dive. So for you, our listener, just to recap the key points, researchers found 14 proteins with different levels in the blood of women with LAM. They confirmed significant changes for 4 of them: fibronectin, VWF, kallikrein III, and plasminogen, showing these interesting imbalances between blood levels and what might be happening in the lung tissue.
Right. And these proteins tie into really core processes in LAM, that ECM remodeling, the cell trafficking, which helps explain things like disease recurrence after transplant.
And then there's that aha moment, perhaps.
Yeah.
This unexpected potential link between all these LAM biomarkers and markers seen in various parasitic infections.
Yeah, that connection, while not the focus of the LAM study itself, is definitely food for thought. It suggests seemingly very different conditions might share some molecular fingerprints.
Which could be a really promising avenue for future investigation, couldn't it?
Absolutely.
It definitely leaves you wondering about the implications. Could this lead to new blood tests? Could targeting these proteins or pathways offer new therapeutic options for LAM patients?
And it prompts that final provocative thought for you to consider. Could digging into these shared biomarkers, this overlap between LAM and parasitic infections, actually unlock unexpected insights into what fundamentally drives LAM? Could it point towards, I don't know, previously unconsidered environmental or maybe even infectious factors playing a role?
That is a powerful question to ponder. If you're interested in exploring this more, we definitely encourage you to look up the original study in PLOS One and perhaps even trace some of those links between the specific proteins and parasites we discussed.
There's a lot more detail out there if you want to dive even deeper.
Absolutely. Thanks for joining us for this deep dive.